Hello again, everyone. My name is Cheryl Boggs, and today we're going to be finishing up talking about photosynthesis. So we'll be discussing the Calvin cycle, environmental conditions and how they affect photosynthesis, as well as mechanisms that certain species of plants have developed through evolution to prevent a process called photorespiration. So just as a quick review from the first module that we talked about, the net overall equation for photosynthesis is 6 carbon dioxide plus 6 water molecules in the presence of light yields glucose, C6H12O6, and 6 molecules of oxygen. So we've already discussed where the water comes into play, which is in the splitting, or when the water is split to replace the electrons in photosystem 2. The light, of course, is there to excite the electrons in the light-dependent reactions, and the oxygen is produced as a result of the splitting of water, photolysis, to replace those electrons. And photosynthesis, of course, before we move on, is it an endergonic or exergonic reaction? Well, overall, photosynthesis is an endergonic reaction. Remember, that means that energy needs to be put into a system in order for it to move forward. And in the case of photosynthesis, light is the energy that's being kind of input into the equation from the sun or from another source. Photosynthesis occurs in 2 stages. We've already discussed the light reactions or the light-dependent reactions in Module 1, and the Calvin cycle is the second piece, the second stage, and that's what we will be discussing today. Again, one more piece of review before we move on is to follow the energy in photosynthesis. So hopefully you remember that the first law of thermodynamics states that energy cannot be created nor be destroyed, but can in fact be transferred or transformed from one form to another, and that's what's going on in photosynthesis as well as in cellular respiration and many other processes as well. So the energy comes into the system through the process of photosynthesis in the form of light, and it is used or converted to produce ATP and NADPH, and that's what we talked about occurring in the light reactions. Okay, and that occurs in the thylakoids, those little green discs stacked on top of each other inside of the chloroplasts. And then that ATP and NADPH is used in the Calvin cycle, which we'll discuss today, which occurs in the stroma, the fluid that surrounds the thylakoids inside the chloroplast, to produce the organic compounds or the carbohydrates that we typically think of as being the products of photosynthesis. So without any further ado, let's move on to the Calvin cycle. So in the Calvin cycle, kind of the broad overview of what happens is chemical energy from the light reactions, okay, that's ATP and NADPH, along with carbon dioxide, which is retrieved from the atmosphere, okay, are used to produce organic compounds. And we typically think of molecules like glucose as what's being produced, okay? And down there on the bottom left of your screen is a schematic diagram of glucose, and that's a good molecule to be familiar with in terms of kind of its general structure and shape. And if you look carefully, you can see it's got 6 carbons, 12 hydrogens, and 6 oxygens. So it's in that classic 1 to 2 to 1 of carbon, hydrogen, and oxygen ratio that we typically associate with carbohydrates. And the Calvin cycle, as I mentioned just a moment ago, occurs in the stroma of the chloroplast. So it occurs in that fluid that surrounds the thylakoids, as you can see there in the diagram of the chloroplast on the bottom right of your screen. So the Calvin cycle involves a process that's called carbon fixation. And what carbon fixation is, it's a term you definitely want to be familiar with. It's the process of assimilating or kind of taking up and making part, taking carbon from a non-organic compound, like carbon dioxide. Okay, we discussed in the first module that to be organic, you must have both carbon and hydrogen. Carbon dioxide does not fit the bill. It's a non-organic compound. And then incorporating it into an organic compound, like a carbohydrate. So that's what you can see there on the bottom of the screen. On the left-hand side is a molecule of carbon dioxide. And through the process of carbon fixation, that carbon is used or is incorporated or assimilated into an organic compound. So if you kind of follow the black here is the carbon, okay? You can see the carbon as it sort of kind of has become part of the carbohydrate. And we're going to talk about specifically how that happens in just a moment. But again, you do really want to be familiar with that term carbon fixation. So it's taking carbon from a non-organic compound and kind of forcing it or making it part of an organic compound. Okay. So this happens the Calvin cycle happens in a few steps, and the first one is carbon fixation, okay, and that's what we've sort of just been talking about. But basically what happens in carbon fixation is we have 3 molecules of carbon dioxide, and those come from the atmosphere, okay, so the carbon dioxide enters the leaf, if we're talking about plants, through the stomata, okay, with the guard cells open. And when those carbon dioxide molecules enter the leaf, right, remember they're stored in those air spaces between the spongy mesophil, they're joined to 3 separate molecules of RUBP, and RUBP stands for ribulose bisphosphate, but you need to know it as RUBP, that it's a 5-carbon sugar, okay, and there's an enzyme called Rubisco. And Rubisco has 2 names. It's also called RUBP carboxylase. But basically what its job is to take these carbons over here on the left-hand side, which are coming from carbon dioxide, and join them up, okay, form a bond between each of these carbon dioxide molecules and a corresponding RUBP molecule. Now, kind of throughout this little module, I've just shown the carbons. So carbon dioxide, of course, is CO2, right? But we're just focusing on the carbons. RUBP has more than just 5 carbons in it. It has hydrogens and oxygens and other molecules, other atoms. But for clarity, I'm just showing the carbons, and hopefully that will be clear. And you don't need to know about all the other kind of atoms, but you do need to understand how this works. So basically what happens here is we have our enzyme Rubisco, and Rubisco is going to join each of these carbon dioxide molecules to an RUBP molecule. And so when that happens, what we end up with is, instead of, as we started, we had 3 5-carbon molecules, right? If we exclude the carbon dioxide molecules, and kind of after this first step is done, what we have are 3 molecules that each have 6 carbons. And so right now, we have a total of 18 carbons. Okay, so hopefully you can see that just by counting the circles on the screen. And we just need to pay attention to those numbers. So we started with 15, right? 3 RUBP molecules, which each had 5 carbons. But then 3 carbon dioxide molecules were added, and those came from the outside, right, from the atmosphere, diffusing in through the stomata. And so the other thing to point out before we move on to the next slide is that the black carbons and the red carbons are exactly the same, but I color-coded them just so we can keep track of kind of where these different carbons came from as we move through the rest of the Calvin cycle. Okay, so here's step 1, carbon dioxide enters the leaf from the atmosphere through the stomata. Rubisco joins the carbon dioxide to RUBP. Okay, that's step 1. This carbon, the carbon from carbon dioxide has been fixed. Okay, it has been incorporated. It's now part of an organic compound. The second step in the Calvin cycle is reduction. Okay, and reduction, the first kind of piece of this is these 3 6-carbon molecules that we just made in the last step in carbon fixation. They're very unstable, and so they're much more stable in terms of chemistry if they are split in half, so if they are smaller molecules. So that's what we see right here. Okay, so we haven't gained or lost any carbons here, but it's basically just been some rearranging that's occurred. Okay, they've just split in half essentially, and hopefully you can see that. Okay. Then the second step is called reduction, and reduction, remember, in terms of like a redox reaction, is gaining electrons. And that might seem a little bit weird at first, right? You're gaining something, so why are you said to be reduced? Well, you have to remember, of course, that to be that electrons have a negative charge, and so if you gain electrons, you're gaining something negative, and so your overall charge is being reduced. And these electrons that are going to be added, or added to our kind of 6 3-carbon molecules, they're coming from NADPH, and hopefully you remember from the first module that NADPH is like an electron taxi. Okay, it's an electron carrier. So it took that excited electron, which got excited away from photosystem 1, and it's shuttling it over here to the stroma to be used for in the Calvin cycle. Okay, so they're going to gain some electrons. Here's our NADPH, if you remember, again from the light reactions, okay? And it's going to donate its electrons to those 6 3-carbon molecules, and then once it has given up its electron, it goes back to being NADP+. Okay? So remember, NADP+, once it's got electrons, an electron and corresponding hydrogen, it becomes NADPH. Once it has given that back up, it goes back to being NADP+. Just like a taxi driver, right? Once it drops off, once he or she drops off their passenger, they're able to take on a new passenger. And so this NADP+ will be basically recycled. It'll shuttle back to the light reactions, pick up more electrons, and then come back. Okay, so this cycle is going on over and over and over again. Okay, and in order to rearrange this, I mean, just think about anything, right? So if you rearrange a room in your house, that takes some energy. And in the same way of kind of rearranging and reshuffling all of these atoms, that takes some energy. And energy, of course, comes from ATP, okay, in terms of where cells get their energy from. So we've got some ATP, and hopefully you remember where the ATP came from. That would be the light reactions. Okay, remember, produced through the process of chemiosmosis when those protons were diffusing through the ATP synthase. Okay, and the ATP is used, okay, producing, and once that phosphate group pops off, to provide that energy, we're left with ADP and phosphate. And again, those molecules can be reused, and they can be kind of forced back together. But that needs to happen through chemiosmosis again. So hopefully you can see that, you know, we need the NADPH and the ATP in order to do this step. So we certainly need to do the light reactions before we can do the Calvin cycle, okay, because we need the ATP and NADPH from the light reactions to run or drive the Calvin cycle. Okay. Oh, well, I just answered my own question right there. Where did the NADPH and ATP come from to do this? They came from the light reactions. Okay. All right, so here's what we're left with. I'm not showing the electrons or kind of the ATP used in rearranging, but again, we're just following the carbon. So here's where we are. We're at step 3 essentially now, okay, or well, I guess we're not quite there yet. So there are now 6 3-carbon molecules, okay, and each of these molecules are called, they have 2 different names. They could be called G3P or PGAL, okay. You may see both terms on the AP exam, so you need to be familiar with them, okay. And if we're just counting carbons, right, remember that we started out the Calvin cycle with 15 carbons. Remember those 3 5-carbon molecules of RUBP that we started with, and now we have 18, okay? So we have a net gain of 3 carbons. If we are talking about, we went from 15, now we have 18, again, as you can see on the screen. So where do those 3 extra carbons come from? Well, hopefully you can see the red ones are the extra ones, okay? And they came from the carbon dioxide, which was brought in through the stomata, okay? So this is where the sugar kind of gets made, and so since we have 3 extra carbons, one of these extra molecules, okay, so one of these molecules is extra, it's going to exit the cycle, and essentially a G3P or a PGAL molecule is basically half of a glucose. If you remember that glucose molecules have 6 carbons, so a G3P or a PGAL molecule, one of these guys can basically afford to be lost because we only started with 15, so we need to end up with 15, okay? Remember, this is a cycle, so cycle implies you have to end up back right where you started, and that's what we'll be sort of looking at here. So we have extra 3 carbons, which is one of these 6 molecules. So one of these G3P or PGAL molecules will exit the cycle and be used, and that's what we showed right there. Okay, so it doesn't matter. I could have picked any of them to leave, but one of them is extra and is going to be used to form half of a glucose molecule. And so when we talk about this, once the Calvin cycle turns twice, okay, and it's actually 6 times, and we'll talk about that in just a quick second, those 2 molecules of G3P, right, the 3-carbon molecule that we just saw kind of leave the group of 6, will combine to form 1 molecule of glucose, okay? And remember that glucose is a 6-carbon carbohydrate molecule or another organic compound. So we typically think of glucose, but the Calvin cycle provides pretty much all of the raw materials, the organic compounds that all organic compounds in the body are, or excuse me, in the organism are going to be made from. So here's our G3P, okay? And notice that I said from 3 turns of the Calvin cycle. So I'm showing sort of 3 turns of the Calvin cycle together because it's much easier to see one of these 3-carbon molecules leaving than seeing 1 carbon leave and 1 carbon leave and 1 carbon leave. So I showed 3 carbon dioxide coming in, and so one G3P coming out. But really that happens kind of one at a time. Okay, so that's why I'm talking about 6 times it would actually need to turn instead of twice. Okay. Once we do it again, okay, 3 more times, we would get another molecule of G3P, just like the first one. Remember, the red and the black are equivalent, doesn't matter in terms of the color. And so what essentially would happen once the Calvin cycle turns a total of 6 times, 6 carbon dioxide coming in, meaning 6 extra carbons can leave, those are joined together to form our glucose molecule. Okay, so just think of G3P or pGal as half of a glucose molecule, that once we have 2 halves, they'll join together to form glucose. So really, even though we talk about glucose being the product, really G3P is actually the product, or pGal is the product of photosynthesis of the Calvin cycle, excuse me. Okay, so that's the second step, reduction, and I'm just showing you kind of how these guys leave, these G3P molecules leave to form glucose. But again, as I've been emphasizing, this is the Calvin cycle, so we need to end up back where we started. And if you remember, we started with 3 molecules that each had 5 carbons, and those were RUBP, or ribulose bisphosphate. So we need to end up back at the beginning, as I just mentioned. And so basically what happens now is the 5 G3P molecules that we have left, remember we had 6, okay, but 1 of those molecules left, 3 carbons left, leaving us now a total of 15 carbons. These guys are going to be rearranged into 3 5-carbon molecules instead of 5 3-carbon molecules. So it's the same number of carbons, we're just rearranging them to get back to RUBP. So where do we get the ATP from to do this? Because remember, anytime you rearrange something, it takes ATP. Well, again, hopefully you remember, it comes from the light reactions. Without the light reactions, the Calvin cycle cannot occur. So ATP is used. Okay, so there we go. We've got 1 molecule of of RUBP. Here we go, our second molecule of RUBP, and a third molecule of RUBP. And if you look back to where we started in step 1, this is essentially exactly what we had to start. Okay, the carbons may be different, right? Some of the carbons that were there originally are now perhaps in that G3P molecule, and maybe now we have some of those carbon dioxide that came in being part of the RUBP, as you see here, with those red carbons now being incorporated. But the key thing here is we're back to where we started. Okay, so what can happen now is if 3 more molecules of carbon dioxide come in, the whole process can begin again, provided that there's NADPH and ATP from the light reactions to run the process. Okay. So again, in this third step here, we started with 5 G3P molecules, which each had 3 carbons, so we had 15 carbons total, and at the end of step 3, we've got 3 RUBP molecules with a total of 15 carbons. We just rearranged them using ATP. Okay? So here's a very simple schematic diagram of what's going on during the Calvin cycle, okay, that we've just been talking about. So we have our RUBP, those 3 5-carbon molecules that are kind of waiting there. Carbon dioxide is kind of brought into the picture and joined to the RUBP by Rubisco, or RUBP carboxylase. Okay, it's the carbon fixation. The NADPH is used, the electrons that NADPH brought to the table from light reactions is used, and then the NADPH reverts back to NADP+. The ATP is used, forming ADP and phosphate, which, not really useful for energy. And what's coming out of the cycle is an organic compound, G3P, pGal, think of it as glucose, all of those things. Okay, that's what's produced. This is a much more complex diagram. It's very clear that you do not need to memorize the names of all of these intermediates. So here's RUBP that I was talking about, okay? And here's G3P that I was talking about, but you don't need to know the names of these other kind of components, and so I wouldn't worry so much about that. But some people find this really helpful in terms of kind of seeing what's going on and where molecules are moving. Okay. So a quick recap of the Calvin cycle. In the Calvin cycle, energy and electrons, which came from the light reactions, right, in the form of ATP and NADPH, along with carbon dioxide, which came from the atmosphere into the organism, into the plant through the stomata, are used to produce organic compounds. okay, like G3P, P-gal, and then eventually potentially glucose. Okay? The Calvin cycle occurs in the stroma, which is inside the chloroplast, which is inside the cells, which are inside the leaves, right? So if we're talking about plants, so just remember the scale is important, okay? Carbon dioxide is required, ATP is required, and NADPH is required in order to run the Calvin cycle. And what's produced are organic compounds like G3P, and those are our products. So we've kind of finished the steps of photosynthesis, so a broad overview of the whole process. The light reactions, which is phase 1, which we talked about in module 1, are responsible for capturing the energy in sunlight and converting it to chemical energy in the form of ATP and NADPH through the use of photosystems, electron transport chains, and the process of chemiosmosis. After the light reactions, the Calvin cycle occurs, the second phase, and it uses the energy that was transformed by the light reactions, okay, the ATP and the NADPH, along with carbon dioxide, which enters the organism through the stomata, to produce organic compounds. So that is photosynthesis. Let's make sure that the equation makes sense, and hopefully if you understand the process of photosynthesis, it shouldn't be hard to learn the equation because you know what you need for each step. So let's talk about it, okay? So light, of course, is required, right? And that's shown over the arrow there. Well, what do we need the light for? Specifically, why is light required for photosynthesis? It excites the electrons in the photosystems during the light reaction. So if you know that light is required, and you know what's going to happen if the light is removed or unavailable. Okay? We need 6 water molecules in order to perform photosynthesis, and hopefully you remember that the water is split during the light reactions to replace electrons that are lost from photosystem 2. Remember, that's called photolysis. 6 carbon dioxide molecules are also required in order to perform photosynthesis, and hopefully you remember that that's where the carbon comes from to make the organic compounds, the G3P, the P-gal, the glucose, that is produced during the Calvin cycle. So the carbon dioxide is required in the Calvin cycle, not in the light reactions. Products, we have 6 molecules of oxygen that are produced. Those are produced during the light reactions as a very fortunate byproduct for us as aerobic organisms during the splitting of water. So when water is split or photolysis occurs during the light reactions, oxygen is released as a byproduct. And also C6H12O6, right? There's our glucose that's produced in the Calvin cycle. It's the organic compound that is ultimately produced through the process of photosynthesis. Okay? Now, it's important to be able to design an experiment to measure how quickly photosynthesis is happening, or the rate of photosynthesis. And so if you understand this equation, hopefully that will make sense, right? So as a reaction occurs, the amount of reactants, or things on the left side of the arrow, should go down, okay, as they're being used up in a closed system, and the amount of products should increase. The things on the right-hand side should go up. There should be more of them, again, in a closed system, as the reaction continues. So just think of an example, right? Let's talk about a table factory, okay? And let's start there. Let's say there's a prescribed amount of wood that's available. Well, as time goes on, hopefully, right, throughout the course of the day, you have fewer pieces of wood in your warehouse, fewer reactants, the things here on the left-hand side of the equation, but you have more tables, right? So you're going to have more products. Again, that's assuming you've got some good workers who are taking your raw materials and making the products that you're looking for. Okay, so the amount of these guys should go down and the amount of these should go up. So most realistically, the easiest ways to measure photosynthesis would be if you were in a closed system, to measure the amount of carbon dioxide that goes down. Okay, the concentration of carbon dioxide should decrease as photosynthesis occurs. In a closed system as well, the amount of oxygen should increase, right? It's a product. So if the photosynthetic autotroph is pumping out oxygen, the amount of oxygen in the atmosphere, again, a closed system, should increase. And then also if you had access to radioactive carbon dioxide, so carbon-14, which is radioactive, then that radioactive carbon would end up here in this carbon in the glucose, and you could measure the amount of radioactivity, so there would be an increase in glucose. So that would, kind of those three ways would probably be the easiest way to measure it, the gases, carbon dioxide and oxygen being probably the most practical. Here's an animation, or a link to an animation that you can take a look at to kind of review your knowledge of photosynthesis and take a look again at kind of where molecules are moving and how things happen. An excellent review for you. Okay. Now, the rate or speed of photosynthesis can vary, and that depends on environmental conditions. So we just talked about how we would measure how quickly photosynthesis happens, right? The increase in oxygen or glucose or the decrease in carbon dioxide. But how fast that happens can really depend, and it can depend on kind of three major environmental conditions. The first, light intensity, temperature, and then oxygen concentration. So we're going to explore each of these briefly. So, in general, if we look at this graph, what we see on the bottom here, we see light intensity, and it's increasing as we move from left to right on the x-axis, and we see the rate of photosynthesis increasing as we move from bottom to top on the y-axis. So hopefully what you're seeing is, right, that as light intensity increases, the rate of photosynthesis does as well, but that doesn't continue forever. So in general, as light intensity increases, so too does the rate of photosynthesis. So photosynthesis happens more quickly under increased light intensity. And the reason why that is, again, if you understand photosynthesis, this will make perfect sense. The more light you have, the more electrons that can get excited, or that are excited in the photosystems, in the light reactions, and that's just going to speed up the whole process. Okay, so the process of photosynthesis will be happening more ate. Okay? However, and hopefully you can see here on this graph, right, it levels off. The rate of photosynthesis levels off at a certain light intensity, and we don't have units on this graph, but that's besides the point. So it increases pretty steadily, pretty dramatically, but then it levels off. It doesn't increase anymore. Now, this doesn't mean that photosynthesis stops, right? If it meant that photosynthesis stops, hopefully you would know what that graph would look like. It would drop back down to nothing, right? And that's not what happens. It still is happening just as fast as it was, at this spot, at this level of light intensity when you get here, but it's not going any faster. And the reason for that is because the photosystems will eventually become saturated. And a good way to think about saturation is, to think about a sponge. If you leave a sponge in a bucket of water for 3 hours and you come back, okay, you can't put any more water into that sponge. It has absorbed all of the water that it possibly could. And so the same thing sort of happens here. The photosystems get so much light and they're excited so often, you just can't increase past that level. Okay, so above that limiting level of light intensity, no further increase in photosynthetic rate will occur. Okay, and that's our light saturation point, right, where it levels off, where it stops increasing. But again, that doesn't mean photosynthesis stops. It's still going very, very quickly, but it's not continuously increasing. If it was continuously increasing, what would our graph look like? It would just continue up, 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 up indefinitely. Okay, and that's not what we see. Temperature. Now the effective temperature on the rate of photosynthesis has to do with enzymes, and enzymes are something hopefully that you're very familiar with. Enzymes are proteins that kind of catalyze biochemical reactions, and in general, and we should have just looked at this graph before I revealed that, but hopefully you see that as temperature increases up to a certain point, so too does the rate of photosynthesis. So they have a direct relationship. Okay. There we go. So as the temperature goes up, the rate of photosynthesis also goes up. Okay? But that doesn't happen forever. Okay? What's going on here at about 30 degrees Celsius? It drops off. This is what we were talking about on the previous slide. Okay? So the reason why it goes faster and faster initially as temperature increases is because molecules move faster, right, at higher temperatures. And this all just has to do with kinetic energy. So as molecules are moving faster, they're more likely to run into or collide with enzymes, okay? And that's going to facilitate chemical reactions. So molecules that are, for lack of a better term, kind of running around faster are more likely to bump into an enzyme and be catalyzed by that enzyme than molecules that are moving more slowly, which is characteristic of molecules at lower temperatures, excuse me. However, once we get past this optimum point, okay, which hopefully you can see right here, it kind of reaches an optimum or a peak level of rate, the rate of photosynthesis decreases. And the reason for that is that the enzymes are denatured. Now, hopefully you remember enzymes are proteins, right? And proteins typically have 4 levels of structure. We've got our primary structure, those amino acids linked together by peptide bonds, and that's the primary structure. Secondary structure, the alpha helix or the beta pleated sheet. The third, the tertiary structure, which is kind of the complex 3D globular shape with the disulfide bridges and lots of other things that are kind of holding that molecule together in a specific 3D shape. And then quaternary structure, which is where you have more than one polypeptide involved in the structure. So at higher temperatures, those intermolecular interactions are disrupted, and so that specific 3D structure that the enzyme or any protein has is disrupted, and it kind of falls apart. And the best way to sort of think about that is, you know, taking your house key or car key and sticking it in a Bunsen burner for 15 minutes. It's going to lose its shape. And so in much the same way, you know, it won't start your car or open the front door of your house any longer. In much the same way, once enzymes lose their shape, they don't have that specific active site that fits the substrate, and so they'll be unable to catalyze the reactions. And that's what when we talk about enzymes being denatured, they've lost their specific 3D shape. Now, we didn't talk a lot about enzymes when we went through the process of photosynthesis, but there are a few enzymes that are involved. We did talk about Rubisco, or RUBP carboxylase. Remember that joins the carbon dioxide to the RUBP in that first step of the Calvin cycle. We talked about ATP synthase, which is that transmembrane protein that spans the thylakoid membrane and allows for chemiosmosis to occur. Another enzyme is NADP plus reductase, which is responsible for adding the electron to NADP plus there at the end of the second electron transport chain in the light reaction. So certainly extremely important that those enzymes have proper structure, and if they don't have their proper structure because they've been exposed to extreme temperatures in terms of heat, they will no longer function. So remember that, always remember that relationship between structure and function. Oxygen concentration is another piece and another environmental factor that affects photosynthesis. So let's take a look at this graph and figure out what it's telling us before we reveal anything. So as we see on the x-axis, the amount of oxygen concentration is increasing, okay, as we move from left to right, and the rate of photosynthesis is also increasing as we move from bottom to top. So what is the effect of increased oxygen concentration on the rate of photosynthesis? Well, it's an inverse relationship. Okay? As the concentration of oxygen increases, the rate of photosynthesis decreases. So the more oxygen there is in the environment, the less quickly photosynthesis happens. And the reason for that is because of something called photorespiration, which is what we're going to talk about next. So photorespiration is, you need to remember that it's not something that's beneficial for photoautotrophs. This is something that they are hopefully trying to avoid. Again, not consciously, but it's not good for them in terms of survival. It's a negative process. What photorespiration is, it's when Rubisco, remember that enzyme we talked about, RUBP carboxylase, joins oxygen to RUBP in the first step of the Calvin cycle rather than carbon dioxide. So we talked about CO2 coming in through the stomata and then being joined by Rubisco to RUBP. And then we go through the Calvin cycle, we get our G3P or our glucose as the product. Well, photorespiration happens when oxygen is joined to RUBP instead of carbon dioxide. And sort of what decides which one is joined is whichever one there's more of. So this is just simple majority rule. If there's more oxygen, oxygen will be joined to RUBP by rubisco. If there's more carbon dioxide will be joined to RUBP by rubisco. Now, the key thing here is photorespiration. When oxygen is joined to RUBP, it prevents the synthesis of glucose and it utilizes or uses up the plant's ATP. So it really is very negative for the plant because it prevents it from joining the carbon dioxide, which would allow it to produce the G3P and thus the glucose, and it also uses up its energy stores, so certainly negative. Okay, here's another way to sort of look at it. If there's more carbon dioxide, rubisco joins carbon dioxide to RUBP and photosynthesis happens and we eventually get glucose. However, if more oxygen is present, rubisco joins oxygen to RUBP and something called photorespiration happens, okay, which means that glucose is not produced. So, photorespiration, why would it be a problem? It's going to be a problem when there's too much oxygen in the environment, and that's not something we're used to talking about. So photorespiration is mostly a problem for plants who are under water stress. Water stress being they're concerned, essentially, or their cells, may not have enough water. Okay? When plants are under water stress, they close their stomata to prevent water loss through transpiration. So remember that the stomata are those tiny holes, typically on the underside of leaves, where carbon dioxide comes in, right, to facilitate the Calvin cycle. Oxygen is released, and water is released through transpiration. So when plants are under water stress, their stomata are going to attempt to prevent the excessive loss of water. They know transpiration is a problem, so they basically shut the door. They shut the stomata and say, we can't afford to lose any more water. Okay? But what are the consequences of that? Well, it also limits gas exchange. The doors are shut, so oxygen can't leave, carbon dioxide can't come in. So the thing is, though, that oxygen is still being produced through the light reactions, right? That water is still being split, oxygen is still being produced, but now it's building up inside of those air spaces, okay? So the concentration of oxygen is increasing inside of the leaf as the stomata are closed. Carbon dioxide isn't entering since the stomata are closed, the door is shut. So as the carbon dioxide is being used up in the Calvin cycle and not being replenished, the concentration of carbon dioxide is decreasing. Okay, so all the carbon dioxide is stuck outside the plant because the stomata are closed. So as the concentration of oxygen increases, okay, remember still being produced through the light reactions, and the concentration of carbon dioxide decreases, the carbon dioxide that was in the leaf is being used up in the Calvin cycle. Again, overall, that's due to the closure of the stomata because of trying to prevent excessive water loss. Photorespiration is favored, and that's a big problem. Remember, doesn't produce glucose and also consumes ATP. Now, some plant species that live in hot, dry climates, okay, where photorespiration is an especially big problem, right? You can imagine that in those dry areas, water stress is going to be a much more common occurrence than in a place like the rainforest, right, where there's plenty of water. So there are some plant species that live in those types of environments that through the process of evolution, through natural selection, they have developed mechanisms to prevent photorespiration. Now, I would be remiss to move on without saying that this is a process that happens in species, right? Evolution cannot happen in an individual organism. So as much as it might benefit an individual, plant to develop one of these mechanisms to prevent photorespiration, that is not how it occurs, okay? So remember that there must be a mutation that occurs, and that mutation, of course, you can't plan a mutation. You can't hope to have a mutation in your DNA, right? But if you do happen to have one and it happens to be beneficial, you're more likely to be able to survive. And if you survive, you're more likely to be able to reproduce. And then more individuals in the next generation would be likely to have that mutation, okay? Especially as ones who don't have that mutation might die off. So, we can have a drought one year, but if the organisms that the plants that live in our environment aren't adapted for that. They can't just, oh, it's going to be a dry year, let's become C4 plants. That's not how it works, okay? You either have the adaptation or you don't, and you're at the mercy, basically, of the evolutionary history of your species, okay? So C4 plants and CAM plants, those are kind of the two kind of major groups here. So C3 plants are kind of the normal ones that we've been talking about. All these plants are normal, but they perform photosynthesis in the way that we have described in modules 1 and 2. So they perform both the light reactions and the Calvin cycle in the mesophyll cells of the leaves, okay, the palisade mesophyll and the spongy mesophyll. The bundle sheath cells of C3 plants do not contain chloroplasts. Now remember, the bundle sheath cells are the ones that are the purple here, okay, that are surrounding the vascular tissue, the xylem and the phloem. So their bundle sheath cells cannot perform photosynthesis because they don't have any chloroplasts. Now, C4 and CAM plants modify the process of photosynthesis to prevent photorespiration. So they perform a slight variation in each, quite a little bit differently than the C3 photosynthetic process, in order to prevent photorespiration, to be able to continue making glucose and using their ATP effectively, not wasting it, but also ensuring that they don't dry out or desiccate in the process. So as a brief overview, C4 plants perform the Calvin cycle, that second step, the carbon fixation piece, in a different location within the leaf than C3 plants do. And CAM plants obtain their carbon dioxide at a different time than C3 plants. Okay, and we'll talk about the details of that in just a second. Both C4 and CAM plants separate the initial fixing of carbon dioxide, okay, which is carbon fixation, from the use of carbon dioxide in the Calvin cycle. So they really separate those two things, where they take the carbon dioxide in and fix it, make it part of an organic compound, and then they use it separately in the Calvin cycle to produce the glucose. They both use an enzyme other than rubisco. Okay, they use an enzyme called PEP carboxylase. PEP carboxylase is able to fix carbon dioxide even though there can be a relatively high concentration of oxygen. So it shows a greater affinity for carbon dioxide than it does for oxygen. Okay, so let's go through this. So C4 plants include corn, sugarcane, and sorghum. Okay, plants that, again, you might think about living in drier environments. Okay, and in this process, basically what happens is, as the carbon dioxide comes in through the stomata, this carbon dioxide is transferred from the mesophyll cells into the bundle sheath cells. The bundle sheath cells are impermeable to carbon dioxide. So basically, once you pump the carbon dioxide into the bundle sheath cells, it can't escape. Okay? And so if you think about it that way, your concentration of carbon dioxide is going to go up. Because if you're basically stockpiling all this carbon dioxide in the bundle sheath cells, you're not going to none of it's going to escape, and your concentration is simply going to go up. And so as a result, the Calvin cycle is favored over photorespiration, because there's more carbon dioxide than there is oxygen, because you are purposefully pumping carbon dioxide into those bundle sheath cells. Now, the bundle sheath cells of C4 plants do contain chloroplasts, and this is a difference from the C3 plants. Remember where we said that they don't. So hopefully you can see that here again. This is it's a different artist, so it's different colors here, but hopefully you can see in the bundle sheath cells, there we go, that we've got some chloroplasts as well present in these bundle sheath cells, which we did not have present in the C3 bundle sheath cells. C4 plants use something called the Hatch-Slack pathway, named after, of course, the scientists who discovered how it works. And here's what happens. Okay, PEP carboxylase, again, another enzyme, right? Think of that ACE ending. It adds carbon dioxide to PEP, and PEP is a 3-carbon compound, and it's found in the mesophyll cells. And you can see that there on the right-hand side in the diagram. Since PEP is a 3-carbon compound and carbon dioxide has 1 carbon, this is going to produce a 4-carbon compound initially, okay? And that's why it's called C4 photosynthesis. That 4-carbon molecule then that's produced is moved into the bundle sheath cells through the plasmodesmata. Remember the little kind of openings between the plant cells that facilitate movement of materials between neighboring cells. And so once it gets into the bundle sheath cells, again, you can see that right here, right? Here's oxaloacetate and then converted to malate, and it's moved into the bundle sheath cells, okay? In the bundle sheath cells, the carbon dioxide is released, okay? As you see right here, and the Calvin cycle happens, okay? And there we get our sugar as per normal, right, our G3P. This Calvin cycle is essentially exactly the same as what we talked about in C3, but it's happening, as you can tell, in a different location. It's happening in the bundle sheath cell. Now, once the carbon dioxide is released from this 4-carbon molecule, right, the pyruvate, which the 3-carbon molecule that's left, and hopefully you've heard of pyruvate before, perhaps in your discussions of cellular respiration, it goes back, right, and is recycled, converted into PEP using some ATP, and then the whole thing can happen again, okay? So taking a look at this diagram, here's a great question. If the Hatch-Slack pathway, as we just discussed, helps to prevent photorespiration, because once you pump that carbon dioxide into the bundle sheath cell, it can't escape, why wouldn't all plants have this adaptation? Well, there's a couple things to notice about this, okay? Number one, again, thinking about evolution, you cannot simply decide to have a certain, biochemical process as part of your makeup, right? I would love to be able to fly to beat traffic to go to work. That's not something that my ancestors passed on to me in my DNA, so I can't do that, okay? If a plant or a plant species did not inherit a mutation for a certain biochemical pathway or the genes to do it, they can't, right? They're at the mercy of the evolutionary history of their species. But the other thing is there is another downside to doing this, okay? If you notice, this conversion of pyruvate back to PEP requires ATP. Now, plants that do C3 photosynthesis don't have to spend that extra energy. Basically, this is a little more expensive of a process to do than C3 photosynthesis. However, if you live in a hot and dry environment, and this is your kind of mechanism to prevent photorespiration and actually produce some sugar, okay, it's probably worth spending that extra ATP. But it's not, in terms of energy, it's not more efficient, okay, compared to a C3 plant, because you need to spend a little more energy to make it happen. Okay, that ATP needs to be spent. So hopefully that makes sense. So two major things to summarize. Number one, if you don't have the genetic mutation to do the Hatch-Slack pathway, you can't. Okay, and secondly, it's a little more costly in terms of input of energy required to perform this process. Cam plants are a little different. They also have an adaptation to prevent photorespiration, but theirs is a little different. Okay? Cam plants include succulent plants, right? Think of plants that store a lot of water, like cacti, as well as pineapples. Okay? Again, living in tropical, warm places. CAM stands for crassulacean acid metabolism, and plants that do this type of photosynthesis open their stomata at night to obtain carbon dioxide and release oxygen. Well, what's the advantage of doing that? Well, typically at night, it's cooler, okay, and not as dry. And so plants that open their stomata at night to get carbon dioxide and release oxygen are still able to do the gas exchange they need to do, right? To get the carbon dioxide they need to do the Calvin cycle and release the oxygen to prevent photorespiration, but they're not going to lose as much water by opening their stomata at night as they would if they opened it during the day. Okay, so keeping their stomata closed during the hottest and driest part of the day helps them to prevent desiccation or drying out. Okay. Now, when their stomata are open at night, the carbon dioxide is converted to an organic acid, okay, as you can see in the diagram there at the bottom right, via the C4 pathway, so much in the same way. Okay, so malic acid is produced, and it's stored overnight. So the carbon dioxide comes in, and it's stored overnight as an organic acid. Now, the carbon dioxide cannot be used during the day, even though it comes in cannot be used at night, even though it comes in at night. And the reason for that is because there's no light at night, right? And hopefully you remember that light is required to do the light reactions And the light reactions produce ATP and NADPH, which are needed to run the Calvin cycle. So even though the carbon dioxide comes in at night, it can't be used at night because there's no ATP or NADPH. Okay? So during the day, when light is present to drive the light reactions to power the Calvin cycle, carbon dioxide is released from the organic acid and used in the Calvin cycle to produce organic compounds. So basically, these take in, CAM plants take in their carbon dioxide at night and then store it until it can be used during the day. Okay. So again, remember that even though the carbon dioxide is taken in at night, the Calvin cycle can't occur at night because the light reactions can't occur in the dark. And the products of the light reactions are required to run the Calvin cycle. Here's an excellent overview of the process of CAM photosynthesis again, just to be able to see what kind of happens at the different times of day. Okay? So, kind of to summarize, both C4 and CAM plants, which are mostly found in hot and dry climates, have evolutionary adaptations passed on to them through their ancestors, which were successful in survival, probably at least partly because they had these adaptations, which originally somewhere came from a mutation, okay? And that helps to prevent photorespiration. C4 plants perform the Calvin cycle in the bundle sheath cells, which are impermeable to carbon dioxide, so once they pump it in there, it cannot escape. Okay? And CAM plants open their stomata at night and store their carbon dioxide until the morning when the light reactions can run. That's the end of our photosynthesis discussion. I hope that it has been helpful to you. Good luck on your AP exam.